Well, good morning, AP Biology students. This is Shelton here, and I'm just going to give you a quick Screencastify to review the main topics of Chapter 9, Understanding the Cell Cycle. And our main focus is mitosis, so cell division, and also making sure that we understand controls over the cell cycle. As far as review material that you guys should be familiar with, you obviously have your notes that I have provided for you. The test prep review book, this is actually going to be pages 59, so it's actually topic 2, page 59 through 64. That would be a good review for you guys to make sure that you look over after you've gone through chapter 9. And then also the study guide that I've scanned for you from the previous version of the textbook. This is actually chapter 12, is the cell cycle. There's a lot of really good practice questions in there in order to prepare for your test. So when we talk about the cell cycle and cell division, I can look at this particular picture here and I can obviously tell that this is a cell that is undergoing cell division. Two big clues is I can actually see chromosomes and I also see spindle fibers. We only see those right at cell division, either mitosis or meiosis. Now today, again, we're going to be talking about mitosis specifically, which is cell division when we're creating identical nuclei. So, and technically, mitosis and meiosis is describing divisions of the nuclei, and then cytokinesis is then the division of the rest of the cytoplasm. Now, reasons why we need to have cell division is we need to make more cells. Cell theory tells us all cells come from pre-existing cells. So when you actually started, we actually had to make gametes. Gametes are cells that can fuse with other gametes to create new genetic combinations. So when you were first established, your genetic composition was established, two gametes fused together in order to create your diploid cell, where you had two of every chromosome. And since then, mitosis has happened trillions and trillions and trillions of times in order to create the multicellular structure that you are. Now, we also undergo mitosis when we need to repair, when we need to grow, tissue renewal. All of that is creating identical cells, and that's what mitosis is. We're always going from, for humans, in our normal somatic cells being the non-gametes. Our somatic cells have 46 total chromosomes. So every time mitosis happens, we create two new daughter cells, each with 46 chromosomes, is what we end up having. Now, if we were undergoing meiosis, meiosis is often referred to as reduction division. In meiosis, what we would end up having is cells that all had half the chromosome information. They'd have 23. Now, since dividing your genetic information is so important, so whether it's mitosis or meiosis, really what we focus on is we focus on the division of the chromosomes. Now, a chromosome is only visible. We can actually see here inside this nucleus of the cell, a chromosome is only going to be visible right at cell division. Chromosomes are just highly compact, dense DNA. It actually binds around proteins. We'll talk about that in future chapters. But when we actually see this kind of formation in a cell, we're actually seeing this condensation of your DNA into chromosomes. Now, otherwise, your DNA is actually in more of an unorganized form. It's unorganized to us. We can't see it under a microscope. But during the normal phases of the cell, when it's not undergoing cell division, then we actually see our DNA in the form of chromatin, where we really can't see a beginning or an end to our chromosomes. Now, when we do talk about a chromosome, it's really important that we understand, so here's a scanning electron microscope of a chromosome, that we understand that when we can see it before mitosis or meiosis, that your DNA has actually already replicated because we have to divide. I mean, if you started out with 46 chromosomes and now you're trillions of cells, and every cell has to have identical copies of those 46 chromosomes, it's actually the DNA replication is a pretty long process that happens inside the cell cycle. So when DNA starts to condense into chromosomes, your DNA has already replicated. And so what we actually see is we see your DNA with its identical copy attached to it, but we don't know what was the original and what's the copy. So what we do know is that we have two sister chromatids on a chromosome that are held together by this complex protein structure called a centromere. Now, this DNA, identical copy of DNA, that's what my sister chromatids are, held together by a centromere, even though it will eventually become 2 chromosomes. This right now is considered 1 chromosome. As long as the centromere is intact, this is 1 chromosome with 2 sister chromatids. Again, the relationship between the sister chromatids is they're identical copies of each other. So if we were doing AP biology math, this cell here has 1 chromosome in it. But this is a chromosome. We see this right at the end of cell division, which we'll talk about in the cell cycle. This is the beginning of G1 of interphase, is what this would be right at the end of mitosis or meiosis. This is 1 chromosome also. So this cell, this would be what our cell looks like and what our DNA looks like right before cell division, where the chromosome is replicated already. As long as that centromere is intact, this is still considered 1 chromosome, but it's now double the DNA content. And then after cell division, sister chromatids are actually going to separate and become their own individual chromosomes. And so this would be what the cell looks like now at the beginning of the cell cycle, again, back at G1 of interphase. So counting chromosomes, but also understanding that there's a doubling, a duplication of DNA during the cell cycle is actually really important. So when I talk about the cell cycle, this is actually a really important diagram for you guys to make sure that you are understanding. We know that all cells come from pre-existing cells. So most cells are going to continue to replicate and copy. Now, not all cells continue to replicate. They're actually going to enter into what we refer to as a G sub 0 phase. They actually don't continue on a cycle. Now, cells that stop at interphase and they don't continue to replicate, examples of those cells would be cells like your neurons. And also skeletal muscle fiber would be other examples of cells. It's really important that the pattern of connectivity between those cells is maintained. And so once those patterns are established, those types of cells do not continue to replicate. Now, other cells are actually created for the purpose of programmed cell death, which is referred to as apoptosis. So the cells like that would be like in your skin. So your skin is constantly generating new cells that will fill up with a protein called keratin and then die. And that creates this protective barrier on the outside that really is important for water retention and also for protecting us from pathogens invading us. So when a cell is first created in the cell cycle, these 2 cells, so remember in mitosis, we are going from 1 nuclei to 2 identical nuclei, and then cytokinesis is now division of the cytoplasm. So we end up with 2 identical cells is what we end up having occur. Now these are cells that are referred to as daughter cells is what that is called. So because we don't know which one was the original, which one was the new one, they're both kind of mixes of new and old altogether. So we're going to track this cell through its cell cycle. And as soon as cytokinesis is done, the nucleus is reformed, my chromosomes have now actually decondensed, I don't know if that's a word, but now gone into chromatin, this cell starts doing its cell job. So it's got genetic material, it has organelles because the organelles were divided through cytokinesis, but it starts doing its cell job wherever it is in your body. Now, it's not very efficient at doing its cell job. It has to grow. And so this is what is happening during this cycle. This is called the G1 phase of interphase. Now, interphase we classify as the normal phase of the cell. Interphase is when the cell is doing its normal job. Under a microscope, we're going to identify a cell in interphase because we can't see chromosomes. We're going to see an intact nucleus with chromatin inside of it. And so that's from a microscope, all we can determine is it's a cell that's in interphase. Now, we'd actually have to measure the DNA content and also look at how the cytoplasm is changing over time to determine if it was G1, S, or G2 phase. So when Sally the cell here is first created, so after mitosis, cytokinesis, she's actually going to start doing her cell job with interphase. But interphase gets divided into 3 different subsections. The first is my G1 phase. And in my G1 phase, this is my initial growth phase. So what this cell has to do is it has to become more efficient at doing its job. So how it's going to become more efficient is making more organelles, making more enzymes, making sure that there's more protein structure in the plasma membrane to bring in nutrients and export waste products. So G1 is going to be this initial growth phase while the cell is actually maturing and becoming highly efficient. Now, what we'll talk about a little bit later today is we'll talk about a checkpoint that exists here. And if this cell has grown enough and is efficient enough, it actually has the ability to produce an extra protein called cyclins that can move this cell into the next phase of interphase in preparation for cell division. Now, this is also where cells will stop if they're in G sub 0. They'll actually stop at this point, like your neurons, your skeletal muscle fiber, and at this point, now they don't prepare for cell division. Now, if this cell, after G1, is going to be replicating, it actually still does its normal cell job, but it starts the process of copying its DNA. Now, your DNA in your genome is 3 billion base pairs long. So this is a really long process. Well, it's not really long if you think about what 3 billion is, but it's going to take, in the fastest cell division we have, it's going to take several hours in order to copy your DNA identically. So this is now my S phase. If I actually measured my DNA content from here to here, my DNA content is going to double because each of those strands of DNA has to create an identical copy, and those are going to coil up into that condensed chromosome with 2 sister chromatids by the time we can see it at the beginning of mitosis. So this is my S phase, and in the S phase, I'm copying my DNA. S is for synthesis, DNA synthesis. I'm creating identical copies of DNA. Now, once I have copied all the DNA, well, now the cell can prepare for a secondary growth phase. The secondary growth phase is making a little bit extra. Because remember what has to happen in cytokinesis. In cytokinesis, the cytoplasm has to divide, and each of these daughter cells has to make sure they have enough organelles and enough material in order to then be able to survive and do their cell job in G1. So in G2, we make a little extra. We make some extra organelles, we make some extra enzymes, extra cytoplasm in order to prepare for cell division. Now there's an additional checkpoint that exists in here to be sure that the cell is ready to progress to cell division. Now, this is what the M phase is. Now, my M phase, the mitotic phase, I'm going to be dividing the cell into 2 identical daughter cells. But it happens in 2 stages. I have mitosis first, and in mitosis, remember, I'm dividing the nucleus, so I get 2 identical nuclei. So again, in our cells, we have 46 chromosomes. That's our diploid number. So that means we have 2 of every chromosome, we have 23 pairs. Well, what we need to make sure that we're having is 2 new cells, each with 23 pairs of chromosomes. So we can't just divvy out those chromosomes like, whichever ones you get, go for it. Every cell has to have an identical set of DNA. The instructions have to be there. So mitosis is a very systematic, step-by-step process that we need to make sure we're just in general understanding the major steps of how that happens. So mitosis technically is division of the nucleus to create 2 identical nuclei. Now, please understand that even though we're creating identical nuclei, a big confusion that ends up happening is students always think that mitosis produces somatic cells and meiosis produces gametes. That's true in animals. This is not true, though, in plants or in fungus, and we'll address that in our next chapter. Just know that mitosis creates identical nuclei. Always, 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 always is what ends up happening. So after mitosis, then I'm going to have cytokinesis. It actually kind of overlaps when cytokinesis begins. And we also need to make sure we're understanding cytokinesis in a plant cell versus an animal cell and making sure we're getting those differences down. Now, let's talk about mitosis a little more in detail. Let's talk about the steps of mitosis. Now, an easy mnemonic to remember for mitosis, there's actually, your textbook's going to talk about 5 different steps of mitosis. I really just need you to know the major events that are happening in the 4 steps of mitosis, PMAT, PMAT. That is always the order of mitosis and the phases of mitosis, where prophase is my first phase, then I have metaphase, but there's a whole bunch of stuff that happens in prophase to get to metaphase. So that's why there's a pro-metaphase that you guys are going to read. But as long as you understand prophase, the events happening there, you understand metaphase, what that looks like, then you'll understand what pro-metaphase is and why there's this significant this 5th step in there. So prophase first, metaphase second, anaphase is third, and then telophase is my 4th and final step that actually overlaps then with cytokinesis. So let's take a look at what's happening during these steps. And if we get them down now, these are still the same steps that happen in meiosis in our next chapter, chapter 10. So when I look at a cell, so let's actually go to just our pictures here. So when I look at a picture of my cell here, I know that this is a cell that is in interphase. And how I know this is a cell in interphase is my DNA is it's an uncondensed form. This is my chromatin. My nuclear envelope around my nucleus is still intact. And also I have these little centrioles. So these little microtubule structures, and this is just in an animal cell. I have these centrioles, and I have this region here called a centrosome that's just packed full of enzymes that are going to eventually help us with cell division. And since my centrioles are still together, my centrosome is just in one location, I have a nuclear envelope intact, and my DNA is in chromatin, I know that I'm still in interphase. Now, what I'm seeing here, though, is the beginning of prophase. My chromosomes are actually starting to become condensed, and I can start seeing my sister chromatids attached together. I'm starting to see my centrioles migrate to opposite poles, and I'm also seeing this mitotic spindle starting to form. Now, this mitotic spindle, these are, again, part of the cytoskeleton that actually chromosomes are going to need to attach to in order to be able to migrate to opposite poles of the cell. But I'm seeing this beginning. Now, what's also going to continue to happen then in prophase, my chromosomes have to separate, so they cannot be contained inside of a nucleus. So the nuclear envelope has to dissolve. It's not dissolving, it's actually being broken down by hydrolytic enzymes, but my nuclear envelope has to be broken down. My centrioles have to migrate to opposite poles, and my spindle fiber has to be completely formed to go from opposite sides of the cell. So all of this has to happen in prophase. Chromosomes condense. I'm going to have my nuclear envelope disappear. Spindle fibers completely form. If that's what you remember, you are good to go about prophase, prometaphase. Now, an additional thing that has to happen in order to get to metaphase is these chromosomes actually have to attach to spindle fibers. And they're actually attaching to special spindle fibers called kinetochore fibers. Now, there is actually in that centromere, that protein complex that's there, each of the sister chromatids has a kinetochore. Now, the kinetochore is actually a big enzyme complex that can actually attach to the special spindle fibers called kinetochore fibers. Now, let's fast forward. It's a long process of prophase and prometaphase. Let's go to metaphase. Metaphase, all of the chromosomes have now attached to the spindle fibers, and they are lined up in the middle of the cell. So I always think of M for middle. My chromosomes line up in the middle, and technically they are lined up in the middle along the metaphase plate, is what it's called. So it's a three-dimensional plane that these chromosomes attach to. Under a microscope, though, we see it looks like they're lined up like second graders at the end of recess. They are lined up and counted and make sure that they are present and accounted for before division happens. So in mitosis, chromosomes just line up single file. Now we're going to see a difference in meiosis of how in this first metaphase, how the chromosomes line up. That's going to be a huge difference that you're going to identify. Now, in metaphase of mitosis, though, chromosomes line up in the middle. They are attached to the spindle fibers, is what we're actually seeing here. Now, once that head count is done, all those second graders are present and accounted for. Well, now sister chromatids actually separate. And each sister chromatid, because remember that one chromosome was two identical copies of itself attached. And once all of the chromosomes have been lined up, anaphase, and I think of anaphase, A for away, sister chromatids start migrating away from each other. And so what ends up happening is the kinetochore, that actual enzyme that was attached to the kinetochore fibers, actually starts eating its way to the opposite pole. This kinetochore is actually munching on the tubulin and actually eating its way to the opposite pole. So in anaphase, my sister chromatids have now separated and they're moving away from each other to opposite sides of the cell. Now, then what happens is once my sister chromatids are actually on opposite sides of the pole, well, now I can start corralling those back up into two new nuclei. So with telophase is my last step of mitosis. And in telophase, two new nuclei start forming. And so with this, these two new nuclei, the nuclear envelope starts surrounding them. And this is also the beginning of cytokinesis. And for cytokinesis, what ends up happening is there's actually these microfilaments that are almost like the strings of your sweatpants that actually just pull in order to just pinch off the cell. And hopefully, not always the case, but hopefully there's an equal division of the cytoplasm contents is what ends up happening when we have cytokinesis. So this is actually cytokinesis in the animal cell where we actually get this cleavage furrow from just those microfilaments actually pulling and squeezing these cells into two cells is what's happening. So if we look at pictures, here's just kind of a picture of interphase where they're using fluorescent lighting and tagged molecules in here to be able to see my DNA and my spindle fibers as far as what's happening. Here I can actually start seeing inside my nucleus, I can start seeing my DNA start to condense. I can see my spindle fibers starting to form. So I know that I'm in some part of prophase. So here also I got spindle fibers. I have my centrioles on opposite sides. I'm seeing my chromosomes more condensed here. So I'm not yet at metaphase, so I'm still in prometaphase. Here is my metaphase. So my spindle fibers are fully formed in yellow, and now all my chromosomes are all lined up in the middle. Now, when you look at a real picture of this, and remember, it's not as pretty as the picture because these chromosomes, they've got their little sister chromatids hanging out there, and they're kind of flapping in the breeze in there when the centromere is now attached to those kinetochore fibers. So you kind of see it looks like worms that are kind of trying to get away, is what happens in metaphase. But they're all lined up in the middle of the cell. Now what I can start to see is anaphase. And so with anaphase, I'm starting to see that movement of the chromatids moving away from each other to my opposite poles. So that's my anaphase. And then I have my telophase where I'm starting to see this division of the cell and my chromosomes are starting to condense back into chromatin at this point in time. So this is my telophase and the beginning of cytokinesis. So a couple things just to double check that we are understanding about metaphase is with metaphase, remember that those chromosomes line up in the middle, but they are lined up on the metaphase plate. So this is this three-dimensional plane is where the chromosomes are lined up in the middle. Also remember that my centrioles in an animal cells have centrioles, plant cells do not have centrioles. But we have two opposite poles of the cell where I'm going to have a centrosome. This is a whole bunch of enzymes that are responsible for building these spindle fibers. And part of the spindle fibers is I'm going to have these special spindles that are called kinetochore fibers, where the kinetochore of the sister chromatids can attach at metaphase. Now I also have another region here that's called an aster. And the aster we can see actually in plant cells and animal cells. And this is where all the spindle fibers are overlapping at the centrosome. And it looks like a star. And that's actually where the word aster even comes from, is star. Now, a couple other things. We take a look at this picture. We used to think, because it kind of looked like these sister chromatids were being reeled in like a fishing line, pulling them to opposite sides. Well, we now know based on experimental design and different experiments and now being able to study it more in-depthly, is we actually now know that there is this protein structure that's called a kinetochore on each of the sister chromatids that attaches to that microtubule, the kinetochore fibers, and it literally just eats itself off to the other side. It's going to break off these little proteins, these tubulin proteins. You don't need to know that, but it's just going to be hydrolyzing, breaking off those as they motor on down to the opposite side. When we talk cytokinesis, do be aware that in cytokinesis, we already talked about this with an animal cell, we've got these microfilaments that can actually just pull the cell into two and create two new daughter cells. So the nucleus has already been reformed, but now I can actually pinch those off and I can actually have now two daughter cells in separating all the cytoplasm, all the organelles and the enzymes. Well, we can't pinch off a wall in a plant cell. So in a plant cell, what ends up having to happen is we actually need to form a new wall in there. This, instead of it being a wall, really popular term plate, whoever named it, terms in cell division here, what actually forms is a cell plate to divide the two plant cells in half. So we build this temporary cell plate, and then this ends up forming a new wall. Now, these are two little small plant cells, but now what can start happening is kind of that reconstruction where more wall is actually expanded out with each of the two cells, but each of these two cells now behaves independently. But we had to form a cell plate where in an animal cell, I can see a cleavage furrow at cytokinesis. So part of your lab will actually be looking at pictures of cells and identifying which phase of mitosis the cells are in. So real pictures, so where you can clearly see metaphase here, I can see anaphase here, telophase here, I can see my prophase starting. I'm not going to ask you guys to identify prometaphase. It's really just important we see prophase, metaphase, anaphase, telophase, PMAT, prophase, metaphase, anaphase, telophase. And we can recognize what those chromosomes are doing. Now, remember, if we're talking mitosis or meiosis, we are talking about cell division in eukaryotes because it's division of the nuclei. We are not talking about cell division in a prokaryote. Prokaryotes don't have a nucleus. So remember this. And also remember that prokaryotes have circular DNA. They don't have linear DNA that forms into rods like chromosomes do. So in a prokaryote, prokaryotes actually have a pretty simple cell division. It's called binary fission. So this is actually identifying how binary fission occurs. DNA in a prokaryote still has to replicate. There's an origin of replication, and we'll talk about this a little bit more when we talk DNA replication because it's circular. There's a clear origin of replication that ends up occurring, and we end up getting two circular segments of DNA. Well, for prokaryotes to then divide, the origin of replication is actually attached to the inner side of the plasma membrane on opposite poles. So we still have opposite poles being established, but I have one ring of DNA on one side, one ring of DNA on the other side, and now I can have a cytokinesis where I just pull those two pieces of DNA apart, and now voila, I get two daughter cells. So I don't have to form spindle fibers, I don't have to worry about nucleus, and this actually leads to prokaryotes being able to replicate very quickly. Some prokaryotes can replicate as quickly as every 30 minutes. They can replicate their DNA, increase all of their cytosol contents, and then divide, which, do the math, that's a whole lot of cells that can actually be created in just a 24-hour time period if replication and binary fission is allowed to occur unaltered. So just kind of keep that in mind. So this is prokaryotes, and then mitosis is in eukaryotes. But then we also have to just be aware of there's a big difference between the two. So we actually have intermediates. In our kingdom Protista, we actually see different types of cell division that are kind of the in-betweeners of binary fission to mitosis. So here, just being aware that I can be dividing the nucleus. Here I've got some different forms of microtubules, but here the nucleus itself isn't quite separated. It doesn't break down like mitosis. Just be aware that we have plenty of evidence of this evolutionary transition between binary fission to mitosis. Mitosis is just highly effective, highly efficient in ensuring that we get identical DNA separated every time. So that is what, through natural selection, has become the norm amongst the domain eukarya. But again, we still have protists that still show some of these intermediate or kind of these in-between evolutionary trends that went from binary fission to mitosis. So lastly, what we need to talk about is we need to talk about control of the cell cycle. This is really important because any cell that is no longer operating under normal cell cycle control, we refer to as a cancerous cell. Something has happened where it's no longer operating at a checkpoint. It might not be ready to divide, but it starts dividing. Well, these are cells that could be potentially dangerous, that our immune system might not be able to combat and eliminate from our body. So cell cycle control is actually a really important identifier. Now, within my cell cycle, there's actually two checkpoints that exist right before cell division. It's a G2 checkpoint, and then there's also an M checkpoint to assure that your chromosomes have divided correctly. But here is actually what I consider my thought process. I consider the G1 checkpoint to be one of the most crucial checkpoints for determining if the cell is going to divide. So remember that my cell starts at G1 of interphase, it's doing its cell job, but here's the G1 checkpoint. And remember, by now, this cell should have become very efficient at doing its normal cell job. Well, this checkpoint exists because inside the cell, there are enzymes that will actually move this cell into the S phase if there is this barrier called cyclin, this protein called cyclin that's been able to be produced in enough concentration to trigger the enzyme, it's called a cyclin-dependent kinase, to now move forward and become activated. So this is what we need to make sure that we are addressing. So here's my checkpoint G1. This is going to determine if this is a cell that continues on to replicate DNA and then the G2 phase. Or is this a cell that's just going to stay there? It's going to stay in G1 until it gets enough energy to make enough cyclin. Or is it a cell that's going to exit the cell cycle and become a G sub 0? So let's take a look at just some of these terms. So here is my G1 checkpoint. Again, this is where a cell might enter the G sub 0 stage where it's not going to replicate at all. But I also then have a G2 checkpoint that determines whether or not the cell is even going to enter into mitosis. And then I also then have an M checkpoint that happens to, in order to make sure that all chromosomes are attached to all the spindle fibers. So that's going to be an important step to make sure that my sister chromatids can separate. Now, how these checkpoints actually happen is, let me just see if I've got the graph here of my cyclin and my cyclin-dependent kinases. So here's just your G sub 0 and your G1 checkpoint that is going to go ahead. Here is then my G2 checkpoint that's going to determine if the cell continues on to mitosis. And then I have my M checkpoint during mitosis. So with my, I don't have a graph here that shows my cyclin buildup. Okay, so let's go back to this one here. What happens at this point? There is, again, an existing enzyme in the cell that's called a cyclin-dependent kinase. In short, that's a CDK. That already exists inside the cell. It's in the cytoplasm that exists. What has to happen is the cell has to be able to produce enough of the protein cyclin. Once enough of that protein cyclin has been produced at high enough concentration, that attaches to the CDK, the cyclin-dependent kinase, and now triggers that enzyme to move the cell forward in the cell cycle. Now, the analogy I'm going to talk about in class is I'm going to talk about the bucket at Sunsplash. This is the analogy that I have for the bucket at Sunsplash is my CDK. And that bucket typically is just there. Now, the only time that bucket actually moves is if enough water builds up inside that bucket. Well, that's my cyclin. So water is the cyclin, the bucket is the CDK. Well, that bucket is dependent on the water to be able to now move and cause action. So my cyclin-dependent kinase, that's the enzyme, that's my CDK. And then cyclin is this protein that has to build up in high enough concentration in order to trigger that enzyme to now start cell cycle actions to move to the S phase, or move from G2 to the M phase, or move from the M phase to cytokinesis. So it's CDKs, that's the generic term, and cyclin is what we're talking about. Now, you will actually have an example called MPF. So MPF is a CDK. That's the example they talk about in your textbook. So that's just one example of a type of CDK that already exists. Now, other controls over the cell cycle is actually density dependency. Cells actually, once they're in a crowded space, well, that's a density dependent inhibition that actually stops cells from dividing. This is actually, density dependent inhibition, you guys notice every time you get an injury, you cut your hand, well, now cells aren't next to each other, there's kind of open space. Well, that actually triggers faster cell cycle action and is going to move that cell on until now all the cell membranes are touching each other, communicating with each other, and now they're in the correct density of number of cells around them. Well, that's going to now slow down the cell cycle. So that's a density dependent type of inhibition. Now, if cells are not operating with density dependent inhibition, that if they're crowded, they keep on growing, well, that's what we refer to as a tumor. So tumors now are cells that are operating outside of density dependent inhibition. They just ignore it and they keep growing, and then a big mass is formed. Now, another type of inhibition that we should be aware of is we should be aware of what ends up happening when we actually have anchorage dependency inhibition. What anchorage dependency inhibition is talking about is the fact that cells have to grow under the right conditions. So, and the right conditions are the correct extracellular matrix in that area. So I should not have a muscle cell, even if I injected like muscle fiber cells inside your bone. Well, that muscle fiber cell, even if it was a stem cell, should not be growing into bone. It's not the right extracellular matrix that triggers it to actually grow into its specialization and its specialized form. So when I talk about anchorage dependency requires that special surface for cell division, that right extracellular matrix, in order to perform. So this is where if a cell actually transforms, its DNA changes, so it's no longer paying attention to any cell cycle controls. It's ignoring CDKs, it's ignoring checkpoints, it's ignoring anchorage dependency, it's ignoring then density dependency. This is where we have types of cancer that can actually metastasize. And when I say that it can metastasize, these are cells that can actually break off and now travel throughout the body and attach to different types of anchors, different types of extracellular matrix, and start growing into other types of tumors. So this is the really dangerous types of cancers is when they spread. So, or metastasize means that they now spread and they're no longer paying attention to anchorage dependency anymore. This is one of the issues with breast cancer, why early detection is really important. Breast Cancer Awareness Month, October. So today's Pink Out Day. So just so you know, actually early detection is key to survival to breast cancer. So what's going to end up happening is the tumor, because this is a cell that is no longer operating under normal cell cycle control, it's no longer paying attention to density dependency. Breast cancer always starts out as a lump or a tumor. Now, sometimes that's deeper into the breast tissue and not as easily detected, but this is where monthly detection should be understood that finding a tumor in this stage is really, really important, and then removing that cancerous tissue right away before it starts invading neighboring tissue, and then before it's actually a cancer that can metastasize, because where the breasts are really close to lymph vessels. Lymph vessels are just kind of the secret passageway that allows cells to get to anywhere in the body. So it's not in the blood, it can go in the blood, but there's going to be more resistance and more difficulty in actually spreading through the blood than through the lymph nodes. So this is also why if somebody is diagnosed with breast cancer, first check is we check to see if the lymph nodes also now have evidence of any cancerous cells. Because once it now metastasizes, that metastasis, these transformed cells can grow anywhere, form tumors anywhere, and this is what we need to make sure we're stopping. We need to make sure that we are catching early detection and we're detecting the cancer right away, right when it's this initial tumor, before it actually has a chance to metastasize. So with that being said, this is your cell cycle, understanding that we are talking about mitosis and cytokinesis, how we create daughter cells, but again, making sure that we're understanding my phases of interphase, understanding what I mean by a checkpoint and where do those 3 checkpoints exist and why, and then what is the basis of cyclin and CDKs for monitoring those checkpoints, and then making sure that you're aware of the events, just in general, not all the details that your textbook talks about, of my stages of mitosis. This is division of the nucleus is what we need to make sure that we are recognizing. So with that being said, make sure you guys read the chapter. Make sure you guys are taking it chunk by chunk. Finish the notes, the FRQs. Make sure you're doing plenty of practice questions in the study guide from the end of your chapter in your textbook using the QR reader, and also just making sure that before you take the test, you could be able to answer the you must knows. So with that, this is Mrs. Shelton. I am out of here, and I will see you guys in